Welcome back to the course Fundamentals of General Immunology, and we looked into the T cell and B cell activation, how this B and T cell activation happens in case of infection and also antigen presentation. Then we looked into the co-stimulation, how this B and T cell co-stimulation can lead to further enhancement of the signaling cascade, which can lead to activation of these cell types. In addition, we also looked into how the signals are getting transduced upon the binding of any antigen or any ligand to the receptors. So let's try to understand further on some of the activation control towards the signaling of the T cell and B cell, and then we'll finish this module finally with the note that the T cell and B cell is activated upon a particular stimulus, and if there is a self antigens which are encountered through the MSC molecules, sometimes we get a tolerance, and that tolerance can lead to, say, suppression of the immune response in order to protect the host cells. So let's try to summarize the whole discussion till now, right from the beginning. So we understood that there are two kinds of immune response which is there in the human body. One is called innate immune response. So this is innate, and then other one is adaptive. And this innate and adaptive is different in terms of their genesis. So innate is one which is present right from the birth. So this is present throughout the life of an individual, and this is present from the birth. That means it is coming from the mother side. Adaptive is the one which we are acquiring in due course of time post exposure. So this is post birth. So this is before birth, and this is after birth. Now these two immune systems which are prevalent in the human body is generally defining the criteria how to eliminate the pathogens. Particularly in the adaptive system, we have two arms, particularly the antibody mediated immunity and the cell mediated immunity. The antibody mediated immunity is what we have learned with the help of antibodies. So mostly the B cell is what is responsible for the production of these antibodies, and there are types of different antibodies which we have discussed. While in cell mediated immune response, we can see that these are mainly done by T cells, and the T cells can be of two types or multiple types, by the way, but generally we are talking about T helper cell and cytotoxic T cell population. Now, so this is in a nutshell till now what we have understood. We understood how these are activated, what are the signal cascade which are present in the cells, which can ultimately leading to the activation of these cell types. So let's move on and try to recapitulate one by one the inbuilt concept of this whole cell types. So let's try to understand this control, activation control, in a detailed way, again in the pictorial form. So once the pathogen is entering through the body cavity into the layer, mucosa, submucosal layer, there are cells which are very specialized in nature called dendritic cell. These cells are engulfing this pathogen right at the site of entry, and these are coming into the lymph node. Now in the lymph node we have these vessels called afferent vessels. So these are called afferent, which are allowing the naive T cell. Naive T cell stands for the cells which are not activated or which are not sensitized with the antigen. So these naive T cells are coming through this into the lymph node area. They are kind of recognizing those antigens which are coming through this dendritic cell population. They are identifying and they are doing some kind of an effector function which can lead to the proliferation of the cell type, the similar kind of cell type which is known as effector T cell population. So this become an effector T cell in the lymph node area, and of course we are talking about the follicular area, the outer side area, and the middle area is what we call cortex area. So this is the follicular area. We are talking about this structure called lymph node. So these are actually a lymph node specific modification of the T lymphocyte, and once this T lymphocyte, which are called effector T lymphocyte, which has all the information necessary to produce some effective immune response, is passed through this tube which is called efferent, Efferent is one which can allow the passage of the effector T cell population to again the site of entry, and once these cells are coming to the site of entry, they are effectively killing the cell mass or specific pathogen which are entered into site. So this is actually a loop mechanism where dendritic cell encounters an antigen, it presents to the naive T cell in the lymph node. The lymph node area is actually the site of maturation of the T cell, and once the T cell are matured, they are going to the site or effector site such as the skin or in the gut, and then doing the effective function to neutralize or clear the antigen from the site. So this is actually a loop, and any deviation in this loop can lead to sustained disease condition or non-effective immune response towards the pathogen. So this is about the lymph node, how these are matured in the lymph node. Second is how these antigen presenting cells are actually doing an activation, proliferation, differentiation in terms of effector and memory T cell. So what we understood is when the antigen presenting cell is effectively displaying the antigen in the form of MHC1 in the CD8 specific T cell population, or in the form of MHC2 CD4 specific population, and we can see here that these are MHC1 molecule with beta 2 microglobulin, and here we can see that these are MHC2 molecule. So this is through 2 and this is through 1. So MHC1 can activate, or 2 can activate the individual naive T cell population. These T cell population, once they are encountered with the pathogen or the antigen, can differentiate and start producing different kind of cytokines. One such cytokine listed here is interleukin 2. Now this interleukin 2 is again bind to its own receptor. We call it autocrine function, where the interleukin 2 can self-activate, help to proliferate the similar kind of cell population in that area, and therefore we can see that there is a clonal expansion. These are happening, everything is at the level of lymphoid organ, and once they are differentiating, they are going to the peripheral tissue area to do function. Now the function could be two ways. One way is either they can do an effector function, effector T cell, or effector CD8 positive T cell, which is called cytotoxic T lymphocyte. And the other way is they can produce and store some of the things in the form of a memory cell. So we will have a memory T helper cell, which is CD4 positive T cell, or we have a effector cell. Apart from the effector cell, we will have a memory cell for CD8. Now, so in general, once we are encountering an antigen, the body system produce multiple cell type. Some cell type can be doing an effective function right at the site of entry of a pathogen, and the other cell type, some of the cell type can store the information in the form of a memory cell. It happens in both T helper cell as well as T cytotoxic cell population. So this is what we have learned till now, that these are controlled in a variety of different way, and let's try to move on and see some other details of this activation cycle. Now once we see that the regulation of cell type is happening, we can understand that these regulations are coming because of the changes in the level of gene expression. Gene expression, when there is happen in case of some stimuli, then we can see that few genes are getting upregulated, few genes are getting downregulated. Some examples are listed here. So in the y-axis, we can see that there are the percentage levels of expression, and these are hours or days. So we can see that in the very early phase, we can see that expression of CFOS that's happening in the very early phases, which is later on converted into IL-2 or CD69, and then later on when it goes to say a day, when we can see that there are expression of IL-2 specific receptor alpha, that's also called as CD25, that's happening. Again, when there is a day or two, we can see that there are expression of CD40 ligands, and finally on day 3, post mitotic activity or the effect of IL-2, we can get a cell division to happen, and therefore we can see the clonal expansion is happening. Some of the cells can do an effector function, some of the cells can do a memory cell, and still some of the cells can do regulatory function. And when it comes to regulatory function, we have discussed that there could be an expression of CTLA4 or PD-1 specific kind of a protein. And if we can parallelly look into what is happening in the cell type, we can say that these are the naive T cell which are having an expression of CFOS, and further once there is an expression of CD69, we can see that these are getting retained into the lymph node, and then further based on the IL-2 expression, these cells are getting proliferated, and then further based on the CD40 and 40 ligand interaction which we have discussed in the co-stimulation, we can see that these are getting amplified to provide an effector function. And finally, after some time, once the clonal expansion happened, when there are effector function which is going on, we can find out there is a fine tune of the effector cell population that's happening when the cells are expressing inhibitory receptor. These are the signals which can inhibit the T cell population in general in human body, and these are studied in detail in the last class, CTLA4 and PD-1. So ideally these are controlled in a multiple way. What we can understood in the outside is the effect. These are controlled in the gene level, post expression of different genes which can define the expression of different proteins, and different proteins can lead to different phenotype. And we can also see that there is a plasticity that is happening in the phenotype, and also the effector function of the cells are different. So this is all about how this activation controls are happening and all, and let's try to understand few more things which are important in terms of regulation of these cell types. So when we discussed about B cell and T cell, and we understood that these are produced in response to an antigen and all, there is a very obvious question that how does a B cell kill a pathogen or eliminate an antigen? So basically, what is the mechanism, how a B cell kill a particular kind of an entering pathogen or an antigen? Similar question goes with a T cell as well. So let's try to understand about the B cell first. So once the B cell are getting activated or differentiated, what we know that B cell can express this antibody on their surface. So the B cells are responsible for production of different kind of B cell receptor, which is otherwise called as immunoglobulins. So these are present on the surface of B cell. Once the B cell are getting differentiated, they can either form a effector cell or can make a memory cells. Now memory cell can store all the information, while the effector cell can produce lot of antibody which are secretory in nature. When we say secretory in nature, that means once the B cell activate, these antibodies which are present on the cell surface are getting detached out and come into the circulation, say plasma or serum, and then they can go to a distant site to do effective function, where the antigen presenting cells are presenting an antigen, or if there is any free floating pathogen which are present in the blood or plasma. That means these antibodies are actually the effector molecules, and therefore the B cell specific response is called antibody mediated immune response, AMI. Now this is one way the B cell activation do a function through antibody. Once the antibodies are produced, they can go to the target site and neutralize the entering antigen or pathogen. The second is by actually neutralizing. So they may not directly do anything, but what they can do is, since these antibodies are in response to the viral specific protein or bacterial specific surface protein, they can mask the surface of bacteria or virus. So what will happen? So these specific protein can go and bind to the cellular receptor to do affection. That means once the virus or a bacteria can attach to the cell surface receptor, they can help the virus or bacteria to internalize. And once the viruses are internalized, then they can do an effect or disease condition. What is happening when we have a circulating antibody? When we have a circulating antibody, these antibody can bind to these receptors which are present on the pathogen. Here we are giving an example of a virus. So let's try to understand that these all antibodies which are circulating is masking this antigen. Once these antigens are masked, then probably there is no binding happening in case of receptor, and therefore these viruses getting neutralized. Virus is no more able to attach to the receptor, and therefore there is no disease as such, and that could happen. Second is, suppose if there is a toxin molecule, then also they can bind to the toxin molecule and neutralize the toxin in the same way what they do for the virus. So they can specifically bind to this toxin molecule and essentially protect the host cell to survive, because toxins are small molecules in terms of their size wise and all. It's a kind of a small protein molecule that could do some kind of an effect in the cell. So these are mostly produced by bacteria. So this can be considered as an example of a virus. This can be considered an example of a bacterial infection. So these are the two ways where the antibody does function. One is directly secreting and circulating into the plasma or serum through the activated B cell population, and the second, they can either neutralize the virus or bacteria present in the site of infection. And finally, they can do a clearance once they are targeting to the pathogen directly, either in the macrophages or any other areas. So essentially, what is output of this whole process is antibody is the effector molecule. We are not talking about any cellular here, although B cell is the generator. So B cell generates the antibody, and these antibodies are coming into circulation to do all the effector function. And finally, we get a sterile condition where there is no pathogen available. If there is a less antibody in the immune response system, or if there is a less antibody in the circulatory serum or plasma, we don't see this effect to happen in a proper way. And therefore, the B cell elimination, B cell specific immune regulation is governed by antibody generally. The next one is a very unique phenomena which happens in B cell only, and that's called class switching. Now this class switching is generally from IgM to IgG. Now let's try to understand this unique phenomena, how it is happening. So we have a naive B cell. Naive B cell stands for the cells which are not exposed to any antigen. These are generally presenting IgM on their surface. So when there is a question that can be asked by anyone that what are the immunoglobulins which are present on a naive B cell, the answer must be IgM. So IgM is the very initial molecule which are present in the naive B cell, and once these are present and encountering an antigen, then this antigen encountering can lead to different kind of activation in a B cell. And once there is a change in the B cell structure, either by binding to the antigen or by specific interaction with the helper T cell, because helper T cell will come with MHC2 specific loaded peptide into this B cell population. Once these are encountered through the B cell, this antigen loaded peptide comes to the B cell, then B cell can go and do a different kind of function. Remember, we are talking about the initial surface molecule to be changed to the different type. So once there is an IgM is now shifting to some other type, and that is what we call class switching phenomena. So initially IgM molecule, once the B cell is getting activated, then we can understand what is happening in the DNA level first. So in the DNA level, we understood initially that where there could be a V chain, D chain, J chain, and then we have a constant region, and this constant region can define what kind of chain it is having. So if we are talking about an IgM molecule, that means definitely there are constant changes of mu type. So this mu type chain can give rise to the IgM molecule. Now if there is a class switching, then this phenomena can be possible only if there is a different kind of recombination event that can happen there. And if there is a class switching, then we can see that VDJ, instead of this, we can see that there is a shuffling of gamma chain instead of mu. So instead of mu, now we can get a gamma, and therefore what we see here is IgG molecule coming into picture. So there is a switch from the constant chain from mu chain to gamma chain, and therefore we can see that the area, so if there is a switching of IgM specific chain to IgG specific chain, that means a mu chain is changed into gamma chain, then we can say that the IgM molecule is now changed to IgG molecule, and therefore what we got is now the surface expression of IgG molecules. So initially the IgM was there and now it is IgG. And because of this IgG is a secretory molecules in nature, we can see that lot of circulating IgG molecules are coming into circulation. They are either in the serum or in the plasma. So this could be in the serum or plasma, and this can do an effective way the function in general. So this phenomena is generally considered as at most important when it comes to diagnosis. So let's try to understand this in perspective of a diagnostic point of view. So suppose this is a y-axis and this is x-axis. So this we can simply say time, and in y-axis we can simply say IgG or IgM types. So once there is an initial phase, we can see that the concentration of IgM is pretty high, and in the later time point we can see that IgG is coming into picture, which is now subsiding to the base level slowly in due course of time. That means IgM is present during the initial phase of infection and IgG is present in the later phase of infection. So we can call this phase as acute, and this can be called as chronic. That means the later part of infection. Acute means a very recent infection. So the presence of IgM in circulation can give an indication of a very early infection or acute infection, while presence of IgG in serum can give an indication of a chronic infection. That means a person infected a little longer period of time, and therefore this class switching phenomena has already happened in those individuals where this antigen encountering happened, the chain got changed into say mu to gamma, and therefore we can see a class switching phenomena, and what we get more amount of IgG concentration in the circulation, which indicates the chronic infection. This can happen in various kind of diagnostic which is available in the market, such as ELISA, which we will see in the later classes. ELISA stands for enzyme linked immunosorbent assay, where the IgG or IgM molecules are identified for a particular pathogen, and once we identify this IgM or IgG, we can say clearly that whether this infection is an acute infection or a chronic infection. So IgG based ELISA or IgM based ELISA is what people are developing to identify acute or chronic infection, and accordingly they can justify the statement because of the known biology of class switching phenomena. So this is all about class switching and importance of G conversion from M molecule. So next is we discussed about how B cell is killing the cell population, and let's understand how T cell is killing the pathogen or eliminating the antigen. And for T cell there are two types of T cell which we will mostly discuss: T helper cell and T cytotoxic cell. So let's try to understand the T helper cell first. So T helper cell is again the cells which can activate the B cell to produce antibodies. So ultimately what we are trying to understand here in T helper cell is actually the antibody mediated immune response which we have understood, but what we understood is only this part. That means we need to activate the B cell somehow to secrete out IgG molecules or IgM molecules. So here we have antigen loaded antigen presenting cell which are coming into picture, and naive T helper cell population is encountering those antigen, and once these are activated, these activated cells can produce a lot of cytokines, and these cytokines can do a lot of functions which are various type of activity that can lead to cellular homeostasis. And apart from this, we can see that the B cell activation can lead to generation of different antibodies. The macrophages can be activated. The cytotoxic T cell population can be activated by different kind of cytokines. So, but mostly when we are trying to understand T helper cell, we are trying to discuss mostly about the antibody mediated immune response. So this is what is generally T helper cell specific immune response. Next is what is happening to the cytotoxic T cell population. So how does this cytotoxic T cell kills a pathogen depends upon the 4 or 5 important molecules which are secreted. One is called perforin, second is called granzyme, third is called cerglycin, and essentially this can lead to a phenomena called apoptosis. So essentially this perforin is actually a pore forming. Perforin means one can perforate into the cellular structure. So these are pore forming protein which can embed themselves into the cell membrane to form a pore, which we will see a little later. And then granzyme is a specific enzyme which can start some kind of a process which can lead to apoptosis. These are pro-apoptotic caspases. These are cysteine aspartate specific caspases, the proteases which can cleave the protein and do some function. And cerglycin is actually a specific core proteoglycan molecule which are again cytotoxic in nature. They can stabilize the granzyme. They can also help in the functioning or proper functioning of perforin as well. So these are 3 molecules which are important for the cytotoxic T cell function, and what we are trying to get achieve at the day after all this phenomena is apoptosis, the clearance of the cell. So let's try to understand the first phenomena, how this perforin, cerglycin and granzyme can do the function. So first the CTL, the cytotoxic T lymphocyte, is identifying the pathogen infected cell which are coming into picture. So these are all same type of expression where MHC1 loaded peptide is coming into the site where cytotoxic T cells are present. They can start secreting a lot of granules such as perforin, and this perforin can especially go into the antigen presenting cell and make a small kind of a pore like structure, and this pore can allow these granules to enter inside the cell. And once these granules are entering inside the cell, they can start cleavage of the different proteins which are present in the cytoplasm, and some of the protein which they cleave can activate the apoptosis, which is called programmed cell death. That means ultimate aim of this whole process is to kill the cell type, and to kill the cell type you need to activate multiple proteins, and one such protein is the proteins which are involved in apoptosis processes. And again, this granzyme is a serine proteases which can cleave multiple protein to start apoptotic process. And finally the cerglycin molecule, which is actually a proteoglycan molecule, can stabilize the cells and also can help to make a pore forming unit such as perforin into a specific kind of a structure so that the granzyme can go inside and do the function. They can also help to protect a granzyme, and they can also ensure the stability and effective delivery of the cerglycin through the pore like structure which is perforin based. So ultimately the cytotoxic T cell population can kill the cell which actually carrying those pathogen. So this can eliminate the antigen loaded cells as such. So this is how the cytotoxic T cell population can kill the cell. So just to summarize the whole process, the B cell can do a function by antibody. The T helper cell can activate the B cell which indirectly produce the antibody, and cytotoxic T cell can directly kill by perforating the cell with the help of some of the chemical such as perforin, and therefore you can get a complete sterile condition in the body system. So till now what we have understood is the activation of all the immune cells and how these can modulate the infectivity in terms of making the environment sterile, how the signaling cascade and the other activation pathways are effective in case of T and B cell. We will try to understand some more details of the immune system in our next module. Thank you.